Good day. Today's lecture will be on fish diseases. The topic of fish disease will be broken down into viral diseases, bacterial diseases, fungal diseases, parasitic diseases, and miscellaneous diseases. The diseases we will cover will be primarily related to North American fish. However, the bacterial, the fungal, and parasitic diseases may be applicable anywhere in the world. First slide, please. First group of diseases we will discuss will be viral diseases. Lymphocystis is an irritable virus. This virus affects numerous species of fish and is presented as prominent plaque-like growth that will lie over the fins, the body, as we can see here. And these are slow-growing proliferative masses. These masses can become quite large, as we see in this case, where they develop a almost cauliflower-like appearance. As we can see, these things usually develop as groups, and these plaques become quite large. They can also be single-cell proliferations, where we have almost ick-like appearance on the epidermis. Of course, this is a proliferative lesion of fibroblast, which means that it can go systemic on a fish, developing these plaque-like lesions over serosal surfaces of the body. Histologically, the lesion appears as a prominent hypertrophy of fibroblastic-like cells. These cells have a very prominent cell membrane, hyalinized cell membrane, with abundant eosinophilic cytoplasm, many blue basophilic granular inclusions with this, and a large nucleus. This lesion may have an abundant inflammation, or it may not. Times when we do have abundant inflammation present with this is a time usually when these begin to regress on the fish. Herpesvirus salmonis, or herpesvirus disease of Salmonids, affects primarily young fry. These fish usually present themselves with exophthalmos, pale gills, and ascites. These fish become sick quite rapidly as the temperature is at a temperature fairly cool temperature, usually about 10 degrees centigrade. The way we protect these fish is usually by raising the temperature of a pond or a run that is involved. Unfortunately, as you raise the temperature on these ponds, we also have a decreased amount of oxygen coming to the fish, which can cause detrimental effects to these young fish at this time. The disease presents itself as prominent necrosis in numerous organs. Organs that are primarily affected are the liver, the kidney, particularly hematopoietic elements of the kidney, as well as the heart, spleen, and any other organs that where we have parenchymal damage of tissue, as we see here, necrosis in the liver. A pathognomonic change that we do see is the development of syncytial cells within the pancreas. If this lesion is present, you should think Herpesvirus salmonis in these salmonids. Channel catfish virus is another herpes virus. This disease is a very serious disease of the channel catfish industry. Again, usually young catfish are affected. These fish present themselves with prominent exophthalmos and ascites. If we look at them after we have opened up their intestinal or their abdominal cavity, what we'll see is enlarged spleen, maybe some serosal hemorrhage, enlarged kidneys, and large spleen. You compare this affected fish versus this non-affected fish, you can see some of the differences that we would see within these fish. Histologically, as this being a herpes virus and a pantropic virus, we can expect to see prominent necrosis of numerous organs: liver, kidney, spleen. We also see hemorrhage and necrosis of the gut, as we see in this picture right here. Numerous parenchymal organs will have necrosis going on. Within this herpes virus, you rarely will find intranuclear inclusions. Epithelioma papillosum, or fish pox, is another herpes virus. This can affect numerous species of fish. However, carp seem to be more prevalent getting this disease. This disease presents itself, like lymphocystis, as a proliferative plaque-like lesions over the skin, the body, and the fins. This disease, unlike lymphocystis, is that it is a prominent epithelial proliferation with no other involvement. As you can see in this fish here, again, it can become quite extensive over fins, anywhere on these fish's body. The prominent proliferation is primarily epidermal, where we have this proliferation of the epidermal cells within these areas. Occasionally, you will find intranuclear inclusions present within the fish, epidermal cells. Rare, but it is quite rare. Infectious hematopoietic necrosis is caused by a rhabdovirus. This disease affects trout or salmonids. These fish were usually, this disease is usually seen in fry, in which we see exophthalmos, sybosides, and fecal cast. Just another picture of the prominent exophthalmos in these fish. These fish will die quite rapidly when the infection occurs. One of the characteristic lesions that you will see in this disease is prominent hemorrhage at the base of the head. Now, other areas may see similar hemorrhage, such as on the fins, as well as serosal surface of the body. But this lesion right here at the base of the head in a salmonid, one should think possibly infectious hematopoietic necrosis. Histologically, what we see is prominent necrosis of your hematopoietic elements. Very little necrosis of other organs. It is this prominent necrosis and complete loss of hematopoietic elements of the anterior kidney is usually what we see and is characteristic of this disease. One lesion that is considered pathognomonic, however, my experience has been, I've seen this in other diseases too, is necrosis of the granular cells. This necrosis of the granular cells will occasionally occur. However, I characterize that lesion as primarily a lesion of stress in a fish and possibly viral cause. So if you see this lesion, think viral disease. Spring viremia of carp is another rhabdovirus. This disease affects carp during the spawning season. Fish will present themselves with prominent prolapse of the rectum and cast material developing, as well as ulcers over the body. Within these ulcers, many times you'll culture known pathogens such as Pseudomonas, Aeromonas from these ulcerated lesions. Fish that die or become quite ill many times will develop petechial hemorrhage over the swim bladder, consequently the name infectious swim bladder disease. This lesion can of course be seen in numerous bacterial infections too. However, if one sees this early in the spring in large adult fish, one should think possibly a viral origin and take appropriate viral cultures. Infectious pancreatic necrosis is a serious disease of your salmonid industry. In fact, this disease is one disease in which eggs are tested for yearly to ensure that the stock is not carrying this disease because of its serious nature. Fish that are presented with this disease many times have serosal hemorrhages, hemorrhage in the pancreatic areas, a prominent gelatinous jelly-like material in the gut, petechial hemorrhage in the mucosal surface of the stomach. Histologically, what we see in these fish is prominent necrosis of the center pancreas. Areas of the pancreas will be almost completely destroyed with a relative abundant inflammatory cells present in these necrotic areas. You will also occasionally see prominent necrosis of the enterocytes of the intestinal gut mucosa. Bacterial diseases are quite common in fish and are usually associated either with a viral disease or stress. The problem with bacterial diseases are that many of these diseases look very similar to one another, and that only culturing is going to be the method in which you're going to be able to identify whether this organism was a pathogen in this fish. Aeromonas hydrophilia is very common. This is seen in numerous species of fish and is usually associated with stress or poor water quality. Fish develop acute septicemias and may die without any lesions at all. Others may present themselves with prominent hemorrhage on fins or with ulcers over the surface of the body. Fish that present themselves with these prominent ulcers usually have survived a fairly lengthy time and may not die immediately of the infection. However, culturing of these areas may give you an ability to find the organism that is causing this disease. Pseudomonas is another common pathogen that is seen in fish. Again, fish may develop prominent ulcerations or hemorrhage at the base of fins or on the fins. Looking at this lesion here, this gross photo, I could not tell the difference between Pseudomonas or Aeromonas, and culturing would have to take place in order for us to make a positive identification as the cause of this septicemia going on in this fish. Vibrio is a disease primarily of saltwater fish. This, like Pseudomonas and Aeromonas, is a septicemic disease where the fish may develop ulcerations on the skin, hemorrhage on various fins, in the eye, as we see here, hemorrhage over serosal surfaces, or prominent necrotic areas within the muscle which may ulcerate to the external surface, giving a furunculosis-like appearance. Fish that have an acute septicemia may die. Fish that happen to survive the acute septicemia may develop either ulceration or these large furunculosis areas within the body of this fish. Edwardsiella tarda, or Edwardsiella septicemia, is a disease primarily seen in catfish and cultured eels, particularly in Asia. Here, fish affected with this disease we'll see many times is septicemia, where we have prominent necrosis or prominent hemorrhage over fins and body surfaces. Catfish may develop also prominent ulcers over various parts of the body. This disease, like Vibrio, can cause prominent little areas of necrosis in muscles and fillet. And when these fillets are flayed out, they can become quite malodorous, which would obviously decrease the quality of a fillet of meat from one of these fish. Edwardsiella ictaluri, or enteric septicemia of catfish, will look very similar. Here, an acute septicemic, we'll see hemorrhage over fins, serosal surfaces of the animal. Prominent areas of epithelial necrosis. Oral hemorrhage, ulcerations. Hemorrhage over serosal surfaces on the body cavity, as we see in this heart. Right here, some serosal hemorrhage. And the characteristic hole-in-the-head disease. This lesion, as we can see here, is developed during the septicemic process of the disease. The septicemia occurs in this fish. The fish survives the acute septicemia. The bacteria lodge in the frontal bones of the fish, erode through the frontal bones, causing this characteristic hole in the head. Consequently, the disease in fish get the name hole-in-the-head disease. This is considered a pathognomonic lesion in catfish for Edwardsiella ictaluri. Edwardsiella salmonicida, or furunculosis, is a disease primarily seen in trout and goldfish. In trout, it's called furunculosis. In goldfish, it's called ulcerative goldfish disease. This disease, like many of the other gram-negative bacteria that we've already discussed, develop a septicemia in the fish. The fish may show no signs at all of an infection, or may develop petechial or ecmotic hemorrhages over the fins, body surfaces. Serosal surfaces, as we see here. And prominent cavitary lesions, as we had seen already in Vibrio and Edwardsiella. These large cavitary spaces can involve prominent areas of muscle in the fish and become quite extensive. In the goldfish develop primarily large ulcerated areas on the epithelial surface, as you can see here and here. Fish that do survive this can have these areas healed over with prominent scarring of the epithelial tissue and deformity of the fish. Some fish will develop an acute septicemia and the fish dying. Histologically, when we look at all these gram-negative organisms, what we usually see is prominent areas of organisms present in parenchymal tissues. These things will lodge in these areas during the septicemic process, causing very little inflammatory response surrounding these. Good areas to look for these things are in the gas-forming organ, the choroid rete of the eye, and the pseudobranch. Yersinia ruckeri is another gram-negative rod that can cause serious problems in your salmonid industry. Here, what the fish do is develop prominent ecmotic and petechial hemorrhages in the oral cavity of these fish. Now, however, although these fish are affected, and this is a characteristic sign of septicemia, they also will develop prominent hemorrhage and petechial and ecmotic hemorrhages on serosal surfaces throughout the body, fins as well as the mouth. Flexobacter columnaris, or columnaris disease, is a disease that affects both the epidermis and the gills. This disease is a disease primarily associated with poor water quality, stress, or viral infections. Affected fish can become seriously involved, as with this fish here, with erosion and loss and necrosis of the epithelial tissues, with the fish essentially being eaten alive by this bacteria. Many times what will present itself, though, is fraying of the fins and occasional epithelial errors and necrosis within these fish. As one can see here, this fish with exophthalmos and ascites is very suggestive that this fish is also infected with channel catfish virus at the same time. One area of the fish that is usually seriously affected is the gills. Given prominent clubbing and proliferation of the secondary lamella, epithelium will proliferate over these secondary lamella, causing prominent compromise of the respiration of this fish. Eventually, these areas will undergo necrosis and sloughing of the lamella. We can see here in this fish here with prominent necrosis of the gill lamella. And histologically, what we'll originally see is prominent epithelial hyperplasia between the secondary lamella. Many times large filamentous mats of organisms will be seen overlying this filamentous, the gill lamella, and then eventually necrosis, and also necrosis of these gills. Bacterial gill disease is primarily what we call this lesion, particularly when it's caused by Flexobacter columnaris, as well as other organisms such as cytophagia and flavobacterium, will cause a very similar lesion that we see in columnaris disease. And of course, Flexobacter columnaris is the lesion for columnaris disease. These bacteria, again, are seen primarily causing disease when we have poor water quality and associated bacterial or viral infections. Here, what we see is prominent clubbing of the gills and necrosis, eventual necrosis of the gill epithelium. Histologically, you cannot identify which organism it is without culturing the organisms, mainly because it has a very similar lesion, a prominent epithelial hyperplasia due to irritation of the secondary lamella and eventual necrosis of these lamella. Renobacterium salmonarum, or bacterial kidney disease, is a serious bacterial disease of the salmonid industry. This disease is characterized by prominent areas of necrosis and inflammation, particularly granulomatous inflammation, involving the kidney of fish, of salmonids, particularly rainbow trout and brown trout and brook trout. Here what we see are areas, white spots in the kidney. You can see a very large area here of infection. And if you look at it a little bit better, you can see these little spots right here in the kidney involved in this section. However, there's another problem with this, is that this may go into the muscles, forming large cavitary spaces within the fish musculature, very similar to Aeromonas salmonicida. It can also cause prominent little blisters on the fish, which can be seen in very young fish, or many times it is seen in spawning trout, in which if you culture these little blisters, you'll get almost pure cultures of this organism. Histologically, what we see is prominent inflammation and necrosis of the kidney. This is primarily granulomatous inflammation, and if we do a gram stain on it, what we'll see is a gram-positive rod intermixed in amongst this inflammatory infiltrate. Tuberculosis can be seen in numerous species of fish. This disease is primarily a lesion of older fish that have been in aquariums for a considerable length of time. Two mycobacteriums, Mycobacterium marinum and fortuitum, are quite common in causing this lesion. One has to remember, mycobacteriosis or tuberculosis in fish can be zoonotic to man, in which we can develop cutaneous ulcers, particularly on the hands or cooler parts of the body, after working with with infected tanks. Consequently, caution must be always taken when working with tanks that are infected with mycobacterium. Grossly, what we usually see is a prominent emaciated fish with areas, you can see the little white spots on this kidney, prominent little granulomas throughout parenchymal and serosal surfaces on this fish. Fish can become quite emaciated and develop large granulomas, as we can see here, developing at the base of the skull, at the, or really near the gills of this fish. And occasionally, it may go into the bones, causing prominent scoliosis development within these fish. Histologically, what we see on these fishes is a central area of caseous necrosis surrounded by abundant numbers of macrophages and other inflammatory cells. If we look at these areas of inflammation, we'll find usually numerous acid-fast positive organisms present within and amongst the granulomas. Nocardia is another bacteria that can cause prominent granulomas in the fish. This organism may form granulomas, as with tuberculosis or mycobacteriosis within fish, in numerous parenchymal organs and serosal surfaces. However, if you find this at the base of the skull of a fish or near the gill arches, this lesion is very a common place to find nocardia. Culture in these areas and looking at and evaluated for histopathology, you'll find long, prominent filamentous organisms present within the granulomatous inflammation, as one can see here, these long filamentous organisms. Occasionally, as in this case, they may be acid-fast positive. Flavobacterium is another granulomatous disease. Here, fish develop prominent white spots over kidneys or prominent nodular development on serosal surfaces or parenchymal surfaces of other organs. When we look at these histologically, what we see, very similar to nocardia or mycobacterium, is prominent central cores of caseal-necrotic debris surrounded by various amounts of inflammatory cells, particularly macrophages. Again, culturing this is the only way that you're going to identify the type of organism that is causing this. Epithelial cystis is a chlamydia-like disease. Here, fish develop prominent little white cyst on the gill epithelium or over epithelial surfaces, as we can see in these cases right here and here. These little cysts do not seem to really affect the fish very severely, unless, of course, they get large numbers of these, where we can have, particularly in the gills, where we may have prominent gill epithelial hyperplasia. Special stains such as the Machiavello for chlamydia-like organisms or rickettsia-like organisms will demonstrate these little cysts within the epithelium, particularly of the gills. Fungal diseases. Saprolagniasis is probably the most common fungal disease seen in fish. This fungal organism primarily affects only the epidermis of the fish, in which we get prominent cotton-like growths over the surface of the body of the fish, which has been recently damaged. This can also affect gills of the fish. Histologically, what we see on affected fish is usually epithelial necrosis of the superficial epithelium, with the organism only observed within the epidermis. Variable amounts of inflammation are usually seen with this lesion. Occasionally, this can affect eggs. And when it does affect eggs, this is a sign of poor water quality and management amongst the eggs as they're being filtered with water. Unfortunately, occasionally, this fungus can become quite aggressive in nature and go into the muscles and cause a great deal of damage within the fish. Brachiomycosis, or gill rot, is caused by Brachomycetes sanguinis. This disease is characterized by prominent infarction of the gill epithelium, the gill lamella, as one can see here in these little red spots on the gills. Histologically, what we see is prominent thrombosis of gill, of the arteries and vessels of the gills, and numerous irregular shaped fungal hyphae present within these thrombotic areas. Within these hyphae are numerous small little round apelospores, which is considered characteristic for this organism. Oops. Ichthyophonus hofferi, or ichthyosporodosis, is another fungal organism, and it presents itself primarily as a granulomatous lesion. One can see prominent granulomatous inflammation in the kidney, around nodular lesions within parenchymal organs such as the liver. When looking at this, one cannot tell this from a granulomatous infection involving bacteria such as Mycobacterium, Nocardia, or Flavobacterium. However, when one examines these granulomatous areas of inflammation and granulomas, one will find prominent endospores present within the granulomatous inflammation. Parasitic diseases. Ick, or Ichthyopterius multifilus, is a very common disease and probably the most common scourge we see in freshwater aquariums. This disease presents itself as little white spots on fish. These little white spots can be seen on the epithelial surface of the fish or may be seen within the gill lamella. These organisms lie or penetrate the epidermis, where they insist and form large trophozoites present. It is during this stage that we develop the large white spot on the fish. As one can see, the epidermis here, they're completely encased subepidermally. So any type of medications that we wish to place in these animals will not affect these organisms. The life cycle is a very simple but complicated one, in which we have to monitor these so we know exactly when to destroy this organism to prevent it from affecting our fish. These large trophozoites will enlarge and grow and then drop off the fish. When they drop off the fish, they will undergo sporulation and develop numerous tomites, which are released from this large tomont. These tomites then will infiltrate into the water and swim in the water until they find an infected fish to infect. The only way we can kill these tomites is by adding our anti-tomite medication, such as methylene blue or malachite green or copper to the solution at that time, and protect the fish by killing these things. The average time it takes for the trophozoites to develop into tomonts and tomites is approximately 3 days. This can be sped up, of course, by increasing the temperature of your tank. Cryptocarians irritans is the ick, a saltwater fish, and it presents itself just like ick in fresh water, as little white spots over the epithelial surface of the fish or on the gills. Life cycle is very similar. Ichthyobota necrotrix, or coccidiosis, is a protozoal disease with the protozoa attaching to the epidermis or the gills, causing prominent irritation of the skin and epithelial irritation with excessive mucus secretion. These organisms, as one can see here, attach to the gills. And what we'll see there is prominent gill hyperplasia within these areas, as well as excessive mucus secretion. So you not only develop a fish that has prominent mucus secretions and giving it a silvery sheen over the fish body surfaces, but also excessive mucus proliferation of the gills. Trichodyna is another protozoal organism that causes excessive proliferation of the, if they attach to the epithelial surfaces or the gills. These organisms, as one can see here, have a very characteristic appearance, a prominent dynine ring with these little denticles present within it. These things attach to the epithelial surface or the gill surface, and if in excessive numbers, will cause increased mucus secretion. This organism is very commonly seen, as we see here, in between gill lamella, and appears many times not to cause problems. However, in times when we have poor water quality, this can become a serious problem. Way you can identify this organism on histologic sections is the prominent horseshoe-shaped nucleus of this organism within the sections. Dinoflagellates can also cause problems in fish. These are very common in the water, and again, in the conditions of poor water quality and excessive proliferation of these organisms, they may cause serious impact on the fish. Here, if these attach to the gill epithelial surface, we can see we can get excessive mucus secretions. They will attach to the gill epithelium. You can see these on these wet mounts here. They attach, they have this prominent chromatophore giving it some color. And if you polarize these, they will become anisotrophic and have this nice shiny appearance, making it easy for you to identify these in sections. Again, you may find these many times in normal tissue. However, when they do proliferate under periods of poor water quality, they can cause irritation and excessive mucus secretions in either the gills or the epithelial surface. Epistylis, or red sore disease, is a disease only seen usually in wild fish. Here you have these prominent barrel-shaped organisms that have a stalk that attaches to the epithelial surface of the fish, causing an ulcer, usually a single ulcer, at the base of the fin. Henneguya, or Sphaerospora, is a disease that is undergoing prominent change right at the moment. And the jury is still out on what is the cause of this lesion, or proliferative gill disease in fish. This disease usually presents itself in new ponds with prominent necrosis and deformation of the gill lamella. The problem here is there are two sets of cysts within the gill lamella. The intraepithelial cysts are usually quite large and rest in the epithelial surface of the secondary lamella. These do not seem to cause any problem with these fish unless we get excessive numbers, where we will see then, of course, epithelial hyperplasia. The serious one is the development of these large intralamellar cysts. In these, we get abundant inflammation and necrosis and deformation of the cartilage within these areas. Currently, the life cycle is believed to involve a mudworm, and they're on the verge, I believe, of splitting these out. The intraepithelial stage being Henneguya, and these intralamellar stages either being Sphaerospora or Aractinomyxoa. Currently, it appears that it's most likely an Aractinomyxoa is the species that causes this intralamellar damage. Myxobola cerebralis, or whirling disease, is a serious disease of our salmonid fisheries, particularly in areas where we are seeing a considerable loss of fish in these areas. This disease involves a mudworm, a tubifax worm, which seems to have a developmental stage of this organism within that worm. Fish ingest either the worm or get it some other means, and they have prominent necrosis of the cartilage, consequently causing these fish to become very deformed, developing this scoliosis. The whirling disease portion of the disease is characterized by this necrosis of the cartilage around the base of the brain. When this collapses on the fish, causing damage to the CNS, we see the fish whirl and swirl around in the tank, consequently whirling disease. Histologically, what we see is degeneration, necrosis of cartilage with abundant inflammation present within these areas. What we can see are occasional small protozoal organisms present, Myxozoan species present within these areas. If we do special stains, many times we'll be able to identify these organisms present within these areas of inflammation. Microsporidium parasites, such as Glugia and Pleistiophora, are very common in fish. These organisms, such as Glugia, will cause prominent proliferation of cells, causing almost tumor-like growths or xenomas present within serosal surfaces of fish. Pleistiophora, on the other hand, will cause prominent proliferation organisms in muscle, but not the hypertrophy that we saw in Glugia. These microsporidians usually are refractile, and when refract to light, you'll see them somewhat glisten. They're also many times acid-fast positive, so that with an acid-fast stain makes them very easy to identify. Coccidiosis. There are numerous coccidial species within fish. Just a few here. Eimeria subepithelialis, Eimeria carpelli, Sardinia. These are very common in fish. Expect to find them when examining the gut of a fish. However, an important thing to remember too is you may, as in we see this kidney, find these in other organs, such as the renal tubular epithelium, or the epithelium of the bile ducts, or the pancreatic duct. If you see this present, don't be surprised, and evaluate the tissue to determine what type of coccidia these will be. Hexamina is another protozoal organism that is seen primarily in salmon that are kept in runs. This organism is a small flagellated protozoa that is a gut that gets into the gut. If large numbers of these organisms get in the gut, we'll see the development of fecal cast and poor digestion, consequently malnutrition in these young fish, and death. Anchor worms can be seen quite commonly. These organisms, they're a copepod in which they swim in the water. Once they find a fish, they will attach to the fish, forming the organism, causing an intense irritation in the epidermis around these fish. Here we can see an actual case of this, in which these organisms attach tightly to the epidermis, and when they do release, they cause these prominent areas of hemorrhage within these areas. Very common. Look for them when you look at fish. You will find these in the water. Fish lice, Argillus species, are occasionally observed. Again, you got to look for them on the fish when looking for these things. These things many times are not as numerous in this as this case here, but can be seen in numerous species of fish. Look for them on fish and you will find them. Again, when you see these things in this large number, when this suggests possibly overcrowding of the fish. Monogenetic trematodes are very common. These two species, Gyrodactylus and Dactylogyrus, are commonly seen attached to the gill surface of the fish, causing minimal, if any, damage at all. However, if large numbers of these organisms are present, we may see some gill epithelial hyperplasia. Diplostomus spathicum, or eye flukes, are a digenetic fluke. Here what we see is the fish acting as an intermediate host for a pisciferous bird. These organisms, these flukes, will migrate to the lens, causing lens opacities, or into the anterior chamber. Histologically, if we evaluate the eye, what we will see are these immature trematodes present within the lens or in the anterior chamber of these fish's eyes. It appears most of the time they do not cause much damage to the eye, other than maybe a cataractous formation. Just another picture of these immature flukes. Black spot disease, caused by Ulifer, is another digenetic trematode. Here, these immature trematodes insist into the epidermis or into the epidermis and dermis of affected fish. These fish develop a prominent granulomatous inflammatory reaction surrounding these areas, developing prominent little black spots, or in some cases white spots, on this fish. Here's a case in which we see prominent white spot development. Histologically, what we'll see is a prominent walling off of these immature trematodes present, and prominent proliferation of your iridophores, giving a characteristic color to these little black spots within the fish's dermal connective tissue. These do not cause any problems with the fish other than cosmetic problems. Thorny-headed worms caused by Acanthocephalus or Pomphorhynchus are very commonly seen, particularly in trout, wild trout. Here, the gut epithelium may be seriously infected with these organisms, causing very little damage at all. However, occasionally these organisms, thorny-headed worms, can penetrate through the mucosal surface, tunica muscularis, to the serosal surface. And occasionally they will rupture out, causing a peritonitis. Very common finding in fish, particularly wild fish, and most of the time causing very minimal, if any, inflammation at all, or problems at all. And ensacids are very common parasites seen in fish. These are a problem primarily for us from eating uncooked fish. These parasites live within the mucosa or in the intestinal tract of fish. When the fish dies, they migrate out into the muscle where they rest until you or I eat this raw fish, and then of course come down with visceral migrans of the stomach and serious problems. Neoplasms of fish. melanomas of the platy-sortel hybrids. These are commonly, this hybrid is an animal model for melanomas in which we see when the F1 hybrid of the platy-sortel cross is bred back to the sortel, we get prominent melanoma development within these fish. It is believed it is caused by a loss of regulator genes within the melanosomes of the development of these melanomas within these fish. They can become quite extensive and involve large areas of these fish. Hepatocellular carcinomas and hepatomas are commonly seen in fish-fed aflatoxic aflatoxins. This was a serious problem one time in rainbow trout. Once they identifying that aflatoxins were the cause of this, we were able to prevent this by eliminating that problem. Development of hepatocellular carcinomas or hepatomas within fish usually suggests toxic liver damage to these fish, possibly due to an environmental contaminant. One can see here prominent nodular development on the liver, and histologic evaluation will tell you whether it is a hepatoma or hepatocellular carcinoma. Stomatopapilloma of eels is a common problem in migrating eels, American eels. These fish develop prominent proliferative growths over the mouth and lips. These are believed to be caused by a virus. However, we have not ever been able to fulfill Koch's postulate with this. These fish seem to do quite well with these things. You can see they develop into quite papillary growths and don't seem to affect the fish. They tend to regress when the fish goes back out to sea. Papillomas of brown bullheads. This is a disease commonly found in brown bullheads. We don't know the cause. Virus disease is suspected. These can become quite prominent on these bullheads. Occasionally, they can develop, as we see here, into squamous cell carcinomas. Again, a virus has been isolated. However, we've never been able to fulfill Koch's postulate. Lip fibromas of angelfish. This is believed to be caused by a retrovirus. It is seen in female angelfish, where they develop this prominent proliferative lesion on the lips. Dermal fibrosarcoma of Royal Pike. This is a retroviral infection. It affects your walleye pike, in which they get multicentric fibrosarcomas developing on the skin of the fish. They do not tend to metastasize within this fish. However, they can become quite large and aggressive in the skin. Lymphosarcoma of Pike is another retroviral disease. This disease is seen in pike in areas of the Great Lakes, particularly areas where the water quality is less than optimal. And what we see is usually ulcers within the skin of these fish, with prominent proliferation of immature or lymphoblastic type lymphocytes, neoplastic lymphocytes within these areas. Schwannomas of the bicolored damselfish are believed to be an animal model for neurofibromatosis type 1 in man. There are several similarities and differences between this disease in these fish and that of man. Numerous species of fish do develop neurofibromas or schwannomas. However, the damselfish has gained this notoriety in that many times we develop these prominent pigmented nodules or schwannomas over the fish. Now, there are many similarities and differences between this in these fish and neurofibromatosis type 1. Both are plexiform in nature. Both develop schwannomas within the fish. However, in man, the pigmented areas usually are not malignant, where in this animal, the pigmented areas can become quite invasive and malignant in nature. It is also felt that in neurofibromatosis type 1, a man, it is felt it is genetically transmitted, whereas in dermal neurofibromatosis, it appears to be horizontally transmitted and not genetically transmitted at all. With that, this is my last slide. I'd like to thank you for your attention.